Hello everyone, my name is Rahul. In the previous video, we had discussed about cell wall. In this video, we are going to discuss about cell membrane. Cell membrane is universal to all cells, that is, it is present in both prokaryotes as well as eukaryotes. The average composition of plasma membrane is such that the lipids, which include all the types of fats, are 40%, that is, phospholipids, cholesterol, and glycolipids. Proteins constitute 58 to 59% and carbohydrates constitute 1 to 2%. In human RBCs, approximately there are 52% proteins and 40% is made up of lipids. Now, talking about the structure of biomembrane or plasma membrane, various models have been put forward during various times. The first model to come into existence was sandwich model or the trilamellar model, which was given by Davison and Danieli in the year 1935. According to this model, plasma membrane is made up of three layers. First is a layer of protein, which is 20 angstrom thick. Second is a layer of phospholipids, which is 35 angstrom thick, and again a layer of protein, which is again 20 angstrom in thickness. So overall, the plasma membrane is 75 angstrom thick. If we have to give a range, the range will be between 75 to 100 angstrom. Now, this phospholipid molecule is an amphipathic molecule. It is an amphipathic molecule due to the presence of two types of parts. One is a hydrophilic head, which is polar, and a hydrophobic tail, which is non-polar. The hydrophobic tail is attached by van der Waal forces. Now this is the protein layer, 20 angstrom. This is again the protein layer, which is 20 angstrom. This is the phospholipid layer seen in the middle. These are hydrophobic tails, and these are hydrophilic heads. So this is the sandwich model or the trilamellar model. The next model to be put forward was unit membrane model, which was given by Robertson in the year 1959. According to Robertson's model, cellular and organelle membranes are similar, are structurally and functionally similar. Different difference is only in the size and chemical structure. So he said that the membrane that covers the cellular molecule, cellular organelles and the cell itself are similar in structure and function, only the difference lies in their chemical structure and their size. However, both these models were rejected because these models could not explain these models could not explain firstly the semi-permeable nature of membrane, and secondly fluidity the fluidity of membrane. So these models were rejected. The most accepted model of plasma membrane is the fluid mosaic model, which is given by Singer and Nicholson in the year 1972. This is the most accepted model. According to this fluid mosaic model, the plasma membrane is protein iceberg, protein iceberg in the sea of phospholipids. Protein iceberg in the sea of phospholipids. Now let us understand what does it mean. It means that the plasma membrane constitutes mainly of phospholipids and proteins. Firstly, we will discuss about phospholipids. Phospholipids, they are the main constituents of plasma membrane. They form continuous structural framework, form the continuous structural framework of plasma membrane. Now studies have showed that plasma membrane or cell membrane, it is composed of lipids that are arranged in bilayer, that is two layers are present of these phospholipids. They are arranged in bilayer such that hydrophobic tails face each other and the hydrophilic heads face the aqueous environment. Now this is the structure of a plasma membrane. Now we can see that this is a phospholipid, this is the head, this is the tail, this is the second layer of phospholipid with its head and the tail. So the tails, they are facing each other, whereas the head on the outside, it faces the extracellular aqueous environment, and the inner head, it faces cytoplasm, again which is mainly made up of water. So the heads, they face the aqueous environment, whereas the tails, they face each other. Now after the phospholipids, the cell also contains proteins. This phospholipid layer provides fluidity to the membrane So plasma membrane is not a rigid structure. There is fluidity due to the presence of these phospholipid molecules because they are rich in unsaturated fatty acids. They are rich in unsaturated fatty acids which are liquid in nature. Which are liquid in nature. Plasma membrane, it is also known as gassy fluid in nature, which means it can flow. The fluid nature of membrane, it is also important in functions like the fluid nature is important for functions such as growth of cell. Secondly, it is necessary for formation of junctions, formation of intercellular junctions, and for movement of substances through the plasma membrane, whether moving inside or whether moving outside. Cholesterol is present in eukaryotic cell membrane, which is absent in the cell membrane of prokaryotes. The cell membrane contains proteins. These proteins are of two types. Firstly, one type of proteins are known as integral or intrinsic proteins. The other are known as peripheral or extrinsic proteins. These are classified on the basis of ease of extraction. Now we can see in the image that this protein, it is present through and through in the cell membrane. So it is very difficult to extract this protein out of the cell membrane, hence it is an integral protein. However, proteins like this, which are present only on the outside or only on the inside, are known as extrinsic proteins and they can be easily extracted out. So the extrinsic proteins, they are superficially arranged. Extrinsic proteins, they are superficially arranged and thus they can be separated easily. However, these integral or intrinsic proteins are tightly bound to phospholipids. These integral proteins are tightly bound to the phospholipids. Some of these integral or intrinsic proteins, they are partially buried. Some are completely buried. As we can see in this image, this is a completely buried integral protein, whereas this is a partially buried integral protein. So these proteins, they can be intrinsic proteins can be partially buried or completely buried. The proteins which are completely buried, they form tunnels, or which are also known as channels. Hence, the completely buried intrinsic proteins are also sometimes known as channel proteins. They are also sometimes known as channel proteins. Now, Apart from proteins and phospholipids, sugar moieties, certain sugar moieties are also present, which are mainly glycolipids, which are mainly glycolipids, and they are used they are sometimes used in recognition of a cell based on a particular sugar. Now, after the structure of plasma membrane, we will discuss about transport. We will discuss about transport across plasma membrane. Now, the molecules that are to be transported, they can be fat soluble or fat insoluble, that is, they are water soluble. Fat-soluble molecules include gases such as oxygen, carbon dioxide, fats, etc. And water-soluble molecules include proteins, ions, etc. So the fat-soluble molecules, they can easily cross the plasma membrane, whereas the water-soluble molecules, which are insoluble in fat, they require the presence of certain channels which enable them to move across the plasma membrane. So transport across membrane, it can be of broadly two types. One is the passive transport, and the other is active transport. Passive transport, it includes osmosis and diffusion. And the second category includes facilitated diffusion. Passive transport, no ATP is required, whereas in active transport, ATP energy is used. In passive transport, substances move from their higher concentration towards their lower concentration according to the chemical gradient, whereas in case of active transport, the movement of substances is against the chemical gradient or the concentration gradient. That is why ATP energy is required. So neutral solutes can pass easily via diffusion along their concentration gradient. Water can pass via osmosis along its concentration gradient. Ions or other charged particles require facilitation by special proteins that form a channel through the plasma membrane, so that these charged particles or ions, they can pass through these channels and enter the cell and enter or exit the cell. So if this is the structure of plasma membrane, substances such as water and other neutral solutes, they can easily move in and out through spaces between the phospholipids. Molecules such as charged particles or ions, they require the presence of intrinsic proteins which will form a channel. Through this channel, the ions such as H+, Na+, K+, etc., they are going to pass through these along their concentration gradient. This is known as facilitated diffusion. This is known as facilitated diffusion. In case of active transport, such as sodium potassium pump, it is an example of active transport of ions. See, sodium concentration is more outside the cell, whereas potassium is present more inside the cell. So these ions, the ion pumps, what they do is they pump K plus into the cell against the concentration gradient, and they also pump sodium outside. Against concentration gradient. So this is how active transport is achieved. Active transport also requires carrier proteins, along with ATP energy. This is the difference between facilitated transport and active transport. That is, in facilitated diffusion, there is no use of ATP, whereas in active transport, ATP energy is used to transport substances in and out of the cell. With this, we finish off with this video. See you in next video.